Assalamualaikum warahmatullahi wabarakatuh and a very good day to all the jury panelists and respective audience. My name is Puteri Syazwani binti Ghazani, student ID 2018292542, will be presenting my final year project research titled The Potential of Biosurfactant Produced by Lactic Acid Bacteria Isolate from Antarctic Seawater for Waste Cooking Oil Degradation Activity. The word oil degradation activity refers to the mechanism that can cause the alteration of the physical and chemical properties of the oil constituent. Part 1. The Introduction. Over the previous years, it was found that the global usage of edible oils such as cooking oil has grown to at least 1 million metric tons per year. According to data collected by Gunstone in 2002, 80% of cooking oil is used in daily food preparation process such as frying, salad dressing, shortening, and spreads. Repeated heating and use of cooking oil in frying starchy foods such as potatoes produces toxic organic compound known as acrylamide, which are very carcinogenic to the environment. In the Antarctic, permitted release of grey water to the environment from the Antarctic Research Station raised the possibility of waste cooking oil spillage. As we all know, the Antarctic environment is very sensitive to pollution and global changes. The use of conventional cleaning such as in situ burning and chemical surfactant can cause intoxication to the environment. Thus, due to the abandonment of natural biological waste from Antarctic animals such as penguin feces and decomposition of sea sponges into the Antarctic seawater, it is believed that there might be presence of lactic acid bacteria in the Antarctic seawater environment. It was hypothesized that the lactic acid bacteria, or also known as LAB, from the Antarctic seawater had the potential to produce biosurfactant metabolite that can be used in oil degradation activity. Part 2. The Literature Review. To give you a bigger view on the environment situation happening on the South Pole, especially incidents relating to oil pollution on the Antarctic Sea, it was found that a recent study by Conway and Peck 2019 had stated that since the discovery of Antarctica, human activities had implicated Antarctic environment. Dire incidents such as the sinking of the supply ship Bahia Paraiso in 1989 near the Outer Harbour at the Antarctic had caused the release of 600,000 liters of refined oil into the Antarctic Ocean. This incident had caused several elderly penguins, amphipods, and sea urchins to die. The hydrocarbon compounds are the major constituent in oil. Polycyclic aromatic hydrocarbon, PAH, oils such as waste cooking oil are very carcinogenic and mutagenic when being exposed to the environment. So, how does introducing biosurfactant to oil pollutant can help in the degradation activity? Well, biosurfactant is a secondary metabolite produced by certain microorganisms and bacteria. Bacteria such as LAB produces biosurfactant, which is surface active agent that is amphiphilic, meaning having both hydrophobic and hydrophilic moieties. In this case, water-loving head and water-hating tail. When biosurfactants are introduced into water and oil mixture, it can reduce the interfacial tension between two immiscible mixture of aqueous and non-aqueous phase of the pollutants, which promotes emulsification activity. Emulsification is the process when the oil is being degraded and converted into small soluble substrate that can be utilized by the microorganism. There are several types of biosurfactant metabolites produced from different microorganisms. For example, rhamnolipid produced by Pseudomonas aeruginosa, trihalloslipid by Rhodococcus anthropolis, glycoprotic by Oceanobacillus species BRI10, and glycolipidic by Halomonas species ANT3B. Next, let's move on to the problem statement of the research. Oil pollution had caused negative effect to the environment, especially in the Antarctic seawater. The release of hydrocarbon oil contaminant to the sea threatens the life and habitat of the aquatic organism. The use of conventional oil cleaning techniques such as chemical surfactant and in situ burning are less environmental friendly and contribute to more harm to the sea environment. Significance of the study. Data obtained from the study can be able to assist on the understanding of using lactic acid bacteria (LAB) isolate to degrade oil pollutants. The finding will also contribute to knowledge on the alternative use of biosurfactant from LAB in reducing pollutants such as waste cooking oil to the environment. There are 3 research objectives that have been established in this experiment. The first one is to isolate and identify LAB from Antarctic seawater sample based on macroscopic, microscopic, and catalase tests. Second is to screen the biosurfactant production by LAB isolates integrating oil waste such as waste cooking oil based on preliminary tests which consist of oil spreading test, surface tension test, and emulsification index test. The third one is to determine biosurfactant mass production by selected LAB isolates. Part 3. Methodology. To accomplish Objective 1, LAB isolation and identification was done as below. 2 samples of Antarctic seawater consortium sample were obtained from the ECO-rented research lab in UPM. These 2 Antarctic seawater consortium samples were O2B and WB5. O2B sample was Antarctic seawater sample taken 15 meters deep where it is considered as pristine, while WB5 sample was taken on top of the surface of the Antarctic seawater. A volume of each sample was transferred and grown into selective media MRSB, which stands for Demand, Robusta, and Sharpe Prof, for 48 hours at 10 degrees Celsius. The sample were then stripped onto the nutrient agar added NaCl media plate and incubated for 48 hours. 17 colonies were then chosen and further subcultured. Next, each 17 colonies of the subculture sample were stripped on Na and MRSA, where the incubation period on Na only took 48 hours at 10 degrees Celsius, while MRSA took 1 month at 10 degrees Celsius. The pure colony growth from each 17 isolate were then observed macroscopically through physical colony formation and microscopically via gram staining and observing it under the microscope. Catalysts were also performed on each isolated colonies by mixing it with a drop of hydrogen peroxide. Next, in accomplishing Objective 2, which was to screen biosurfactant production by each LAB isolates, 3 types of preliminary tests consisting of the oil spreading test, surface tension test, and emulsification index percentage calculation were done. First, for the oil spreading test, the biosurfactant was obtained from each isolate via centrifuge at 8,000 rpm for 1 minute. 100 microliters of biosurfactant was placed on the middle of the oil surface layer where the spreading of the oil formed and the diameter of the clearing zone were measured. Next, for the surface tension test, the biosurfactant from each isolate were obtained same as the previous method before. The surface tension of the biosurfactant were then measured using pendant drop method on the VCA 3000 machine. Now, for the emulsification index test, the biosurfactant obtained, same as the method before, were mixed and tested with 3 different types of oil, which were waste canola oil, waste palm oil, and waste cooking oil. Each tube containing the biosurfactant waste oil mixture were vortexed for 3 minutes. After setting aside for 24 hours, hence the name of E24, the emulsification index percentage were measured and calculated using the formula height of emulsification in centimeter divided by total height of mixture in centimeter times by 100. For accomplishing Objective 3 in the determination of biosurfactant mass, biosurfactant sample obtained same as the previous method before were filtered and treated with HCl, which stands for hydrochloric acid, and then were mixed with 2:1 ratio of chloroform and methanol for separation. After vortexing each mixture for 15 minutes, the sample were dried in the oven for 24 hours at 80 degrees Celsius. The mass of biosurfactant produced by each isolate were then measured using the formula mass of plate after drying minus mass of empty plate. Part 4. Results and discussion. 17 colonies were isolated from O2B and WB5 samples. However, the results shown based on table 4.1, only 7 out of 17 colony isolated show positive result for the colony growth in the lactic acid bacteria selective media MRSA and MRSB. MRSA is in agar form, MRSB is in broth form. There are some isolated colonies that have the potential to grow on the MRSA media, such as O2BA, O2BE, and O2BG. So it can grow mildly on MRSB on the graph. It was estimated if given 1 month to incubate, it can possibly grow on the MRSA since the colony isolated might have a slower growth rate. Referring to this table, the status column colored red indicated absence or slow growth of bacteria on the selective media. The isolate that were marked red was removed from the experimentation and omitted. Meanwhile, the status column that is colored green showed the positive result indicating that the isolate were able to grow on the selective media. Only 7 positive result isolate are shown: O2BD, O2BF, O2BJ, WB5A, WB5C, WB5D, and lastly WB5E were then proceeded with the LAB identification. Next, on LAB identification, based on table 4.2, all the 7 isolated colonies show circular form, raised elevation, entire margin, translucent opacity, and creamy white color colonies. All the 7 isolate were gram-positive bacteria and produced negative result for catalase test, which aligned to the characteristic of lactic acid bacteria. However, each isolate has slight different microscopic morphology when observed under the microscope. As we can see in figure 4.2, O2BJ, WB5C, and WB5E had short rod-shaped morphology, while the others were long rod-shaped. Some of the LAB on WB5D also had Y-shaped rod characteristic, indicating that some of the isolate were from different species of LAB. Now, moving on to the result for the screening of biosurfactant production by each LAB isolate in all screening tests. It was found that LAB from colony O2BD had the widest screening zone with diameter of 1.25 cm. Meanwhile, LAB from colony O2BF and WB5A had the smallest screening zone with diameter of 1 cm. O2BD biosurfactant had the highest ability for lowering the surface tension of oil, hence resulting higher curing zone. But the effectiveness of both of the highest and the lowest curing diameter isolate are similar. This is because when statistically tested with one-way ANOVA Turkey test, there were no significant difference between O2BD against O2BF and WB5A. Next, on the surface tension result, it is best known that the lower the surface tension it had, the better it is at entering and lowering the surface tension force of higher surface tension molecules. In this case, the lower the surface tension of the biosurfactant, the better it is at lowering the surface tension of oil and water, allowing the mixture to mix, thus promoting emulsification to occur. In this experiment, the lowest surface tension value was from O2BD with 34.09 millinewton per meter. Meanwhile, the highest surface tension value was from O2BF with 45.64 millinewton per meter. When tested with one-way ANOVA Turkey test, there was significant difference between O2BD and O2BF. In the emulsification index test, the percentage rate of each biosurfactant for different isolate to be able to emulsify and degrade oil were tested on 3 different types of waste oil, which were waste cooking oil, waste canola oil, and waste palm oil. For the emulsification index of biosurfactants against waste cooking oil, WB5A and WB5E have the highest emulsification percentage with 50% respectively. Meanwhile, WB5C had the lowest emulsification rate with 32.32%. However, when tested with one-way ANOVA Turkey test, there were highly significant difference between WB5A and WB5E against WB5C. On waste canola oil emulsification, the highest emulsification index for waste canola oil degradation was from WB5D with 47.77%. Meanwhile, the lowest emulsification index for waste canola oil degradation were from O2BF and O2BJ with 40% respectively. When tested with one-way ANOVA Turkey test, there was significant difference between WB5D against O2BF and O2BJ. Last but not least, when each biosurfactant were tested against waste palm oil, the highest emulsification percentage for waste palm oil degradation was from isolate WB5E with 47.78%. Meanwhile, the lowest emulsification rate for waste palm oil degradation was from WB5C with 33.33%. When tested with one-way ANOVA Turkey test, there was significant difference between WB5E and WB5C. Lastly, on the result for the determination of biosurfactant mass produced from each 7 isolates, highest biosurfactant production mass was recorded on O2BD with mass of 1.1 mg. Meanwhile, lowest biosurfactant production mass was from WB5A with mass of 0.2 mg. When tested with one-way ANOVA Turkey test, there was moderate significant difference between O2BD and WB5A. Part 5. Conclusion and Recommendation. It can be concluded that among 17 isolates, 7 isolates were identified as LAB based on macroscopic, microscopic, morphology, and catalyst tests. Moreover, out of 7 isolates screened, O2BD produces the best result in oil spreading tests and surface tension tests. Isolate WB5A and WB5E were the best emulsifier of cooking oil waste. Isolate WB5B was the best emulsifier for waste canola oil, and WB5E was the best emulsifier of waste palm oil. This shows that different isolates give different effect on different types of waste oil, as the viscosity or pH of the oil can affect the emulsification rate. Lastly, the best biosurfactant producer based on mass was isolate O2BD. For the recommendation of future studies, molecular level identification needed to be done in order to identify and determine the species and strain of the LAB isolates. Optimization also needed to be done on the Antarctic lactic acid bacteria isolate to determine the best conditions such as pH for the isolate to perform and produce biosurfactant. Glycolipid studies also needed to be done to determine the type of biosurfactant being produced. For biosurfactant extraction, the use of rotary evaporator to efficiently remove solvent are recommended. Use enclosed tensiometer for better surface tension measurement, minimizing environmental disturbance and false reading. And lastly, increase trial reputation of the experiment for better data collection and accuracy. These are some references that I use for guidance in my research. We have come to the end of my presentation. That is all from me. Thank you.